The study of protein-protein interactions provides an understanding of the regulation of many biological processes. Here, we demonstrate a procedure described initially by Y, John, Shu and co-workers in 2007, where the authors had used BiFC in combination with FRET to validate the tripartite interaction between three protein molecules. However, we have included fluorescence lifetime measurements in this procedure to substantiate FRET measurements.
To demonstrate a tripartite interaction, we have chosen a MADS box protein which is known to homodimerize. Furthermore, it has also been validated in-house to interact with a calcium sensor protein referred to as C protein in this protocol. The M and C proteins interact in the cytoplasm. In this technique, two proteins are tagged with split YFP proteins. Their interaction results in the reconstitution of functional YFP that act as a FRET acceptor to the third interacting partner, which is tagged with CFP acting as a FRET donor. Start with the amplification of the coding sequences of the genes of interest, that are M and C genes in our case, by PCR, and clone them in appropriate entry vectors. Validate clones that are selected on the antibiotics containing plates by restriction digestion and DNA sequencing. Mobilize the reconstituted CDSs from entry clones to the destination vectors. All the vectors used in this experiment are listed in Table 1. Finally, transform Agrobacterium GV3101 cells with the destination vectors by electroporation.